Muy bien.
Venga, perfecto.
Muy bien. Good afternoon, and thank you again for attending the webinar from the SEM. Today we have the webinar from the molecular neurobiology group that is coordinated by Alberto Pascual Bravo from the Instituto de Biomedicina de Sevilla. Thank you, Alberto, for the work the spectacular work that you are doing as a coordinator. And if you want, you can introduce our speaker today.
Thank you, Mar, and welcome everyone to this session. So it's my pleasure to introduce you, our speaker today, Irene López Fabuel. Irene is actually working in Valencia as a Ramón y Cajal Fellow. She has been previously working with another interesting group in Spain. Probably all of you know the team of the Bolaños team in Salamanca, where she did the PhD studies. And over there, she did a really interesting work that she will be presenting today, pretty well published, and I think very adequate to the SEM meeting, because it's a connection between a disease and the metabolism. So I think it's really interesting. And now she's doing also a really interesting work in another top group in Spain with Isabel Fareñas, no? That is really interesting as a place to be formed with. And I think I will stop now the presentation, and I let you, Irene, to start with your presentation. Thank you for accepting the invitation, and the floor is yours.
Thank you, Mari, Alberto. Okay, I think you are seeing the presentation. Okay. Thank you for the presentation. And the work that I'm going to tell you about is our last paper that we do in the laboratory of Juan Pedro Bolaños, called Aberrant Upregulation of the Glycolytic Enzyme PFKB3 in CLN7 neuronal serial lipofuscinosis. Okay, as Alberto told you, we did this work in the Juan Pedro Bolaños lab. That is a laboratory focused on neuroenergetic and metabolism. And Marina García Macía was also a very important part of this work. We started to work in CLN disease or patent disease because we form part of an international consortium called BATCure that was funded by the European Union in a Horizon 2020 project that tried to find therapeutic options for patients and families with CLN3, CLN6, or CLN7 disease. It was involved 7 European countries with 10 different groups, and also with the collaboration of the Association of Patent Disease Family. But what are really patent disease? This is the common name of neuronal serial lipofuscinosis, and they were first described at the beginning of the 20th century, and they are caused by mutation in 13 different genes. These genes give the name to the disease, so we can find CLN1 disease, CLN7 disease, CLN3, and they cause the disease in autosomal recessive inherited form, although we can find one of them that is a dominant form of the disease. These proteins are mainly involved in lysosomal function, and we can find them within lysosome, endoplasmic reticulum, at cytosol, and also one of them is found extracellular. They should share some common function because the phenotypic hallmark of the disease is quite similar. So they are involving essential cellular processes and also developmental processes. And the common hallmark of all of them is the disrupted autophagy that leads to the accumulation of autofluorescent material called lipofucsin in the lysosomes. We can find them within neurons and also other cell types in the body. It is formed by protein and also by lipids. In a normal brain, we are unable to see the accumulation of this autofluorescent material. This is a cerebellum, but in the brain samples of CLN7 model, we can find the accumulation of these accumulations of lipofucsin. When we observe this under electron microscopy, this is the normal aspect of these accumulations within the cells, within neurons in this case. The phenotypic symptoms of the disease is the visual failure and also epileptic seizure, reduced mental capacities, and also motor function. And in all of the cases, they lead to premature death. In the case of childhood, that is the main form of the disease, that when it affects children, it has a very rapid progression. In the adult forms of the disease, the disease tends to be milder, but also affects in a very important way the quality of life of patients and also families. We started to work with CLN7 disease, that is one of the late infantile form of the disease. The onset of the disease starts between 1 to 2 years to 5 years, causing the death between 6 to 18 years. So it's a very devastating disease for families and also for patients. It has a very rapid progression, and with visual deterioration, seizures, ataxia, speech impairment, loss of fine motor skills, and lead to cell death, death of the person. Sorry. CLN7 is a lysosomal transmembrane protein, but we don't know exactly its function. Normally, a recent paper published last year said that it is a chlorine channel in lysosomes, but its function is not really clear at the moment. We used a mice model, although it had been described near 35 to 38 different mutations. Seems that all of them cause a loss of function in this protein, in CLN7, and we are using a genetic model that is caused by the deletion of exon 2. And one of the characteristics of the disease in the mice is the hyaline clasping. That is, that animals cross their legs and they are unable to open them. In this model, the disease progresses quickly, and it says the same pathological hallmarks than humans. So for these reasons, we select this model to start to work with it. And I said you before, this Batten disease or neuronal cytolipofuscinosis has a common marker that is the autophagy impairment. So first of all, we wanted to know if primary cultures of these neurons have this characteristic. And when we culture these neurons with lysosomal inhibitors, we observe that in normal conditions, we have a flux of autophagy market. What this means is the autophagy is working properly in wild-type neurons. But in the case of CLN7 knockout neurons, we were unable to observe this flux. That is meaning that autophagy is not working properly in neurons. But they are right now a metabolic group, so we wanted to know how were the powerhouse of the cells that are mitochondria affected in this condition. And when we make this experiment looking for the abundance of mitochondrial proteins, like the subunity of mitochondrial ATP synthase or AHP60, we observe that in wild-type neurons, we have the same flux. So it seems that mitochondria is working properly through mitophagy. But in the case of CLN7 knockout neurons, we don't observe this flux. So it seems that mitophagy is impaired in CLN7 knockout neurons. And when we observe how were neurons in brain cortex of these mice at the beginning of the biochemical markers of the disease, like is 2 months of age, or in the late stage of the disease in mice, that is 8 months, we observe an enlarged mitochondria and also the accumulation of this, lysosomal material within neurons at 2 months and also 8 months. So seems that mitochondria is affected and we have enlarged mitochondria in this mice model. And when we observe how were these mitochondria working in this model, we observe that they were not working properly. We have a reduced basal respiration, reduced maximal respiration, what means that we have a mitochondrial dysfunction in this model. What we know is that neurons in normal condition, work that we did before at the lab, tend to assemble complex 1 into supercomplexes because neurons are cells highly dependent of oxidative phosphorylation. And this highly assembled of complex 1 into supercomplexes leads to a reduced production of mitochondrial reactive oxygen species in the cells. So maybe the assembly of mitochondrial electron transport chain is affected in CLN7 knockout neurons. And if this is occurring, maybe like was occurring in astrocytes, the increased abundance of free complex 1 could be leading to an increase in mitochondrial reactive oxygen species. So when we did these kind of experiments in a CLN7 knockout neuron, that is mitochondrial isolation, blue native gel electrophoresis, followed by in-gel activity of complex 1 and electroblooding, we observe that we have high abundance of free complex 1 in CLN7 knockout neurons, leading to an increased production of reactive oxygen species, mitochondrial reactive oxygen species in these neurons. So what we know right now is that in CLN7 knockout neurons, we have an impaired mitophagy leading to enlarged mitochondria, and the electron transport chain in this mitochondria is also affected, passing from a supercomplex assembled form of complex 1 to high abundance of free complex 1, leading to the production of mitochondrial reactive oxygen species. So in the lab, we have a genetic model, a genetic mice in which we can reduce the production of mitochondrial reactive oxygen species. That is called mitocatalysis because it has the catalase fused to a mitochondrial epitope, leading to the introduction of catalase within mitochondria. We have these conditional mice, and we thought that maybe crossing our CLN7 knockout model with mitocatalysis model under a specific neuronal promoter leading to the production of recombinase, we could reduce the production of reactive oxygen species within a CLN7 knockout in a CLN7 knockout model in neurons. So we generate these crosses and we obtain from different mice with the characteristics that we wanted to study, and we observed that in this model we were able to reduce the production of mitochondrial reactive oxygen species within neurons. So it was working properly in this condition. And when we observe how were mitochondria at the cortex in neurons, we observe that we were able to reduce mitochondrial area, we were able to reduce mitochondrial area and mitochondrial length, and also reduce the crystal opening in this mitochondria. And this led to a reduction of the biochemical hallmarks of the disease, that is the accumulation of lipofucsin in the cortex. We did also in cerebellum and hippocampus, and also a reduced abundance of subunit of mitochondrial ATP synthase, that is also another biochemical marker of the disease, is accumulation. So we observe that the increased abundance of mitochondrial reactive originating species could be contributing to the phenotypic hallmark of the disease, and the reduction of this mitochondrial production could be improving this disease, the phenotype of the disease. But also, we were a metabolic group and we wanted to know how this mitochondrial dysfunction could be affecting the metabolism of neurons. Cells use glucose through glycolysis, but also through PPP, pentose phosphate pathway. And what we know is that neurons prefer to use glucose in the pentose phosphate pathway. And what we observe is that our CLN7 knockout neurons have an increased levels of glycolytic flux affecting or reducing the PPP flux. So it seems that neuronal metabolism was really affected. Neurons prefer to use the PPP to generate reduced potential. And we know that the enzyme PFK-AB3 produces the allosteric activation of PFK1, and this enzyme regulates the glycolytic flux. So we thought that maybe PFK-AB3 could be affected in CLN7 model. And what we know is in normal conditions, neurons don't have high levels of PFK-AB3, and when we overexpress PFK-AB3, we increase the glycolytic flux in neurons and reduce the PPP activity. So maybe this was what was occurring in CLN7 knockout neurons. And when we measure PFK-AB3 protein levels in brain cortex and also in neurons, we observe that we have an PFK-AB3 accumulation and also an increase in PFK-AB3 activity, but we don't have a modulation in the mRNA of PFK-AB3. And what we know is that in normal condition, in neurons, the complex APC-CDH1 targets PFK-AB3 for proteasomal degradation. So maybe PFK-AB3 is stabilized in CLN7 knockout neurons. And in the lab, previously what described the signal cascade that leads to PFK-AB3 stabilization. And normally, the high levels of intracellular calcium lead to the activation of calpain. Calpain proteolyzes P35 to P25, and P25 binds to CDK5, leading to the phosphorylation of CDH1. In this condition, the complex APC-CDH1 is not formed, and this leads to PFK-AB3 stabilization. So we studied how this leads to the neurodegeneration in a context of excitotoxicity, and we thought that maybe this was occurring in PFK in CLN7 knockout model. When we measure intracellular calcium levels, we observe an increase in intracellular calcium in the CLN7 knockout neurons. And when we don't regulate calcium level with BAPTA, that is a calcium chelator, we observe a reduction in PFK-AB3 protein and also a reduced activity of glycolysis. In the case of the levels of P25, we observe that in CLN7 knockout neurons, the levels of P25 were higher. And when we inhibit calpain with MGL, we observe that we could downregulate the levels of P25, leading to a diminished abundance of PFK-AB3. The next step was to corroborate how was the activity of CDK5 and P25 complex. When we when we knock down CDK5, we observe a reduced abundance of PFKB3. And in normal condition, with a phospho assay, we observe that in CLN7 knockout neurons, we have high abundance forms of phospho-CDH1. So it seems that this signal pathway is occurring in CLN7 knockout neurons, leading to PFKB3 stabilization. So in CLN7 knockout neurons, we have a downward alteration of normal metabolism, leading to an increased flux of glycolysis, diminishing the abundance of PPP in these cells. So we thought that maybe modulating PFKB3 activity could be a valuable target for CLN7 disease. What we used was a highly selective inhibitor of PFKB3 that is called AZeta67. And when we treat primary cultures of neurons with this inhibitor, we observe a reduced activity of PFKB3, leading to a reduced flux of glycolysis in these cells, improving mitochondrial OXPHOS activity, and leading to a reduced neuronal cell death measured by the active form of KSPACE3. Like it was working in an in vitro model, we treated animals with injections of AZeta60C daily in the lateral ventricles during 2 months. And when we studied how were the histopathological markers of the disease, we observed that this treatment was able to reduce the accumulation of lipofuncin and also the accumulation of Cmax in the neurons, in the brain of these animals. And also we were able to prevent the hyaline clasping of these animals due to the treatment of PFKB3 inhibitor. So it seems that with our work, we were able to define 2 different points of actuation to trying to reduce the CLN7 phenotype, that were the downregulation of mitochondrial ROS production and downregulation the PFKB3 activity with a selective inhibitor of PFKB3. And this work was published last year in Nature Communication. Here you have all the information if you want to read it properly. And I said before that in normal condition there is no treatment for this disease, but 1 year ago, 2 years ago, we published a paper in collaboration with Diego Medina at the CIPF in Nápoles. And in this paper, we describe a novel hallmark of the disease, that is the accumulation of HEB3 that we can detect with cigatoxin. And we observe that this is an important hallmark of the disease. We have the accumulation of HEB3 in cerebellum, hippocampus, and cortex of this animal. And we use this characteristic, this accumulation, for the development of high-content screening, trying to find new valuable targets for the treatment of this disease. We use a library of AVA-approved compounds. We use 1,280 drugs, and we define 9 compound hits that could prevent the accumulation of this HEB3 in CLN3 and CLN7 model. Here you can see how tamoxifen, that was one with the highest reduction of the accumulation of HEB3, was able to reduce the accumulation of cigatoxin in these cells. So, and also in neuronal progenitor cells derived from patients with CLN7 disease. For this reason, we and the wave tamoxifen, this activity of tamoxifen was not due to the activation of estrogen receptor, because if we add tamoxifen to cells without estrogen receptor, we observe that tamoxifen was also able to prevent the accumulation of GB3. But what we know that is that tamoxifen was acting through CIPF, leading to its activation at its nuclear translocation to the nucleus. So what we do was inject twice per week tamoxifen to CLN7 knockout mice, and we started to study how were the histopathological markers of the disease. And we observed that the tamoxifen was able to prevent the accumulation of GB3 in the cortex and also in the cerebellum, and also prevent the accumulation of lipofunctionin, ECMAX, and also the microgliosis associated to the disease. And also it was able to prevent the hyaline clasping for these animals. So in this work, what we do was to highlight a possible therapeutic role for this disease, that is tamoxifen. And thank you for your time. If you have any question.
Thank you, Irene, for this nice presentation. So we have time now for questions, and I think you can use the chat to write down the questions so we can read to Irene and she can answer. So I don't see right now any at the chat at Zoom, and I don't know, Mar, if there is anyone
Yes.
at YouTube.
We have one question from Marina Garcia Macia. She says, Irene, great job, beautiful explanation. I have a question. Do you think that the FB3 inhibitor may improve autophagy or lysosomal biology? Thank you. The inhibitor, do you think that is improving autophagy or lysosomal biology? Do you think that is making something in this pathway?
I can't remember. I think that we showed that it was improving lysosomal function, autophagy, and it was also not preventing the accumulation of mitochondrial growth, so it's working by a different way.
Yeah. Because what you see is that the substrates are already accumulated, no, and you cannot inhibit this accumulation when you add the inhibitors of the autophagy, no?
Yes.
In the disease, no? What you see is the accumulation of the substrates, no?
The substrates of autophagy.
Yeah.
Yes, we observe the accumulation of mitochondrial proteins and also autophagy markers.
Okay.
Irene? I don't know, just by looking to the images that you were showing at the beginning, no, with the lipofunctinosis signal and everything, that remember a lot to me to the fluorescent labeling that appear in all brains in wild type. So, you know, I don't know if you have been working with all brains from mouse, but you have a lot of autofluorescence in those, and you need to treat them to do a nice staining and everything. Do you think that that is related with accumulation of those products, and that can be a failure also in autophagy, or in related what it is something that is being accelerated by the disease, and that is also taking place with time in the normal brain?
The accumulation of lipofunctions occurs during aging, so it's normal to see this, but not in the same extent that we have with working with CLN7 models.
Okay.
So it's normal, the accumulation of lipofunctions. What is not normal is the accumulation of the subunit C of mitochondrial ATP synthetase. That is a specific hallmark of the disease.
Okay. Okay. And the connection between the disease and the break of the super complexes in the mitochondria, do you have any idea what that can be happening there? How is the molecular link between the lysosomal problem and there is any protein that could be involved that is accumulated or not well processed that could be leading to the separation of the super complexes?
No, probably it's a consequence. It means if you have an impaired autophagy, you are not degrading properly mitochondria, so you are accumulating dysfunctional mitochondria that could be signaling producing mitochondrial ROS. What we know, or what is accepted, is that high levels of mitochondrial ROS disrupt supercomplexes formation. So if you have high levels of mitochondrial ROS because you have impaired mitochondria, you are affecting also the assembly of supercomplexes. And if you have this and oxidative phosphorylation is not working properly, cells need to obtain energy by one way. What they do is use glycolysis to survive, and it leads to neurodegeneration.
Okay.
So, and do you think that you can do something like, I don't know, maybe by molecular glues trying to put close one complex to the other and increase the performance of the functioning of this chain, or not? Any approximation in this way or not?
Yes, the group of Lucas Corrano published a paper in which they describe that OPA1 reduces cristae opening, and when you have the cristae closed, is more prone to form supercomplexes. So affecting the structure of the mitochondria could lead to better performance of supercomplexes in the cells. And we also overexpress, during my thesis, we overexpress one subunit of complex 1, and doing this, we favor the assembly of complex 1 into supercomplexes.
And had you tried to use any CDK5 modulator to treat the disease or not?
No, we haven't do this. We use only mitocatelase and the PFK3 inhibitor. But we haven't used CDK5 inhibitor.
So how do you sorry, Alberto, tell.
No, it's okay. So I was thinking in tamoxifen treatment for the patient. Do you think that could be possible, or that is quite complicated because tamoxifen has in long term has probably also not unwanted effect on the patients. So do you think we will need a chronic treatment to the patient with tamoxifen? Or?
We don't know. I think there is one project working on it, but we have started way to test if tamoxifen is a good treatment for the disease, because at the moment there is no treatment for the disease. There is only a symptomatic treatment, and a possible valuable target for the disease is really important for the families and for the patients. So we think that we have started an important way to know the answer to your questions.
But Irene, if there is no treatment for these patients, have you started a clinical trial or something like this too?
I think that the collaboration is working right now in this way.
Yeah.
But at the moment there is no treatment. One group published an adenovirus overexpression of CLN7, and seems that is working, but I don't know how they will finish in a clinical trial.
Yeah, but in the case of tamoxifen, as Alberto says, there are some side effects that could be negative, no? But when the disease is so is devastating, no? So maybe you can use it as the first alternative that is for the treatment, no?
Correct. Yes.
Yeah, it's really a good result. Yeah.
And regarding the tamoxifen too, maybe I don't know if there is any other question. No, so far, no.
Not so far.
No. So, what could be the target of tamoxifen? Because you say it's independent of estrogen receptor.
TFEB.
Sorry.
TFEB. It's TFEB. It
TFEB.
improves lysosomal function.
Okay.
And lysosomal biogenesis. And for this reason, I think that it improves the disease because it makes the lysosomal work better.
And could you have other drugs that will be also altering this pathway or improving by this way? Because now that you have localized the pathway that is acting where tamoxifen is acting, maybe you could search for other therapeutic alternatives that are also improving.
Maybe. We need to test.
Okay.
And Jose Castaño asks, what happens to astrocytes? Other neurons are affected in your models? Sorry.
What we know is that the levels of CLN7 in astrocytes are very, very lower than the levels of CLN7 in neurons. We did this. And in CLN7 disease, what occurs is an astrogliosis and a microgliosis. And we thought that this is caused by neurodegeneration, but we haven't studied how is the metabolism affected in CLN7 knockout astrocytes, because in neurons it induces glycolysis, but astrocytes are very glycolytic cells. So taking into consideration the reduced abundance of CLN7 protein in these cells and thinking in they are glycolytic, we haven't focused because what occurs in the disease is neurodegeneration at the beginning.
Actually, neutrons' main energy supply is lactate and not glucose. If I am correct, no? Yeah. Maybe this is the question. Some clarifications that has made Jose Castaño. Neurons' the main energy supply is lactate and no glucose, and maybe this is the difference, no? And one curiosity, because you have said that tamoxifen is inducing an increase in the lysosomal activity, no?
Lysosomal biogenesis.
Yeah, and do you think that some of the drugs, for example, those that are inducing senescence, such as palbociclib, that in the case of senescence induction, there is also an increase in the lysosomal activity? Do you think that you can use such type of drugs for treating the disease, or have you tried?
I think that they could be a valuable target, but I think that they should test yet, because if you have an autosomal lysosomal impairment, I think people should test how a lysosomal modulator will be affecting the disease, and I think there is not positive results with this. I haven't read anything.
This could be interesting. So thank you very much.
See, if there is no more questions, also I will say thank you again to Irene. A really interesting story. I hope you will continue one day. I don't know. You are really working in a different subject right now, no? But I imagine Pedro and the other people in the team will continue with the story. So thank you again, and I hope everyone enjoyed the webinar.
Thank you. both of them, Irene and Alberto, and thank you all the audience also for being with us. And remember that we have a new one, webinar in some weeks that will be properly announced. Thank you very much, Alberto and Irene, for accepting this invitation.
Thank you.
Bye.
Bye-bye.